Okay, so I would like to thank the organizers for organizing this webinar and allowing me to present the safety strategy that we use at the Procter & Gamble company. And we use this for ensuring the safety of our dietary supplement ingredients, especially those of botanical origin. So as a disclaimer, I want to just say that I work primarily on products for the United States marketplace. So I will use the term dietary supplements often. However, the principles that I am presenting for safety will apply regardless of the regulatory definition of the product category. So in my presentation today, I will give you a brief overview of our overall approach to botanical safety science, and I'll highlight the importance we place, especially on deep analytical characterization of our raw materials, and showcase some of our approaches that we use to fill data gaps. Some of those are tools that we are looking at as part of the Botanical Safety Consortium. And then I'll wrap up with a brief summary and a few conclusions. So the development of dietary supplements, including botanical-based dietary supplements, typically mirrors a process that's very similar to other product categories like OTC drug products, for example. Early on, there are elements of exploration, followed by experimentation, and finally production and entry into the marketplace of your final product. So product safety evaluation has to follow along in parallel, and we start very early with hazard identification and risk assessment early in the development cycle, and then we transition later towards market surveillance, or what we call pharmacovigilance, once it's in the marketplace. So product safety will span the entire product life cycle, from upstream design and due diligence to in-market safety surveillance. So for the remaining portion of my presentation, though, I'm going to really talk primarily on the significant amount of work that we put into preclinical safety in assessing the botanical raw materials, and how we approach potential data gaps that we might identify in the process. So we typically think of product safety as a three-pronged approach. First is objective safety. This includes hazard characterization and exposure considerations, which are the core pieces of the overall risk assessment. So we also consider history of safe use, and we do think about traditional use as well. But it has to go beyond that traditional use, and it must include evidence for use in diverse populations and a consideration of potential sensitive populations. So as stated here, it's not if a botanical is safe, but how much and for whom that needs to be determined. We also take into consideration regulatory safety, and this includes requirements to market in a specified region. So are we talking about, certain warnings like specific child warnings for products containing iron, or child-resistant closure requirements on packaging for gummy or soft chew forms, which is typically the situation in the United States. It's just an industry expectation that those products will be in child-resistant closure. And then there's nutravigilance or perceived safety issues. So this relies on consumer habits and practices, trends, co-use type scenarios, And it also includes potential misconceptions, misuse, and abuse of a particular product or ingredient. So we take that into consideration as well. Now I'm going to move on to talk a little bit about that analytical characterization work that we do. Our preclinical safety begins and is so dependent upon a thorough botanical characterization using state-of-the-art analytical approach. So ensuring botanical identity, as you've already heard in the previous talks, is critical in meeting quality challenges and being ever vigilant for potential adulterations or contaminations of botanical raw materials. However, for the toxicologists, it is increasingly important for us to use this information in our botanical safety assessments, but it can also impact efficacy, of course. In many cases, a three-tiered approach is needed for complete identification of botanicals. So this could include visual methods, analytical methods, as well as DNA methods. So botanical extracts present obvious challenges for the analytical chemist. There are multiple isomers, co-eluting compounds, lot-to-lot variation, processing variations, stability issues, and I could go on and on and on. This can also include limited or inaccurate literature data available on phytochemical constituents attributed to a particular species. So at Procter & Gamble, I am very fortunate to have colleagues in our analytical group that we really consider that a core capability that we have here. And they utilize a split stream approach that allows for full identification and quantitation of botanical raw materials. They use UV vis plus tandem high resolution mass spec and a charged aerosol detector, or CAD, that allows us to have both qualitative and quantitative information about our botanical materials. So a lot of analytical horsepower goes in to phytochemical constituent characterization. And the output that is ultimately given to someone like myself, the toxicologist, looks a lot like this as a report. This example is for grapeseed extract. And ultimately, all of that analytical work that they do, they hand it off to me, and I'm really focused primarily on this one column here. I want to know the amount of a particular constituent per dose that's going to be used in the product, because that's what I'm going to use in my risk assessment. So now I'll talk a little bit about how we use that identification information, constituent information, to do our safety assessment. Now, in the interest of time today, I can only present a single case study, but I can give you a reference that talks about several other case studies and how we've used this information. So we've developed and published on a tiered approach that encompasses both in silico and in vitro approaches to identify safety data gaps and inform on the potential need for additional toxicity studies. Today, I'm only going to focus on this tier 3, which is particularly useful in situations where, say, your project formulators want to use a higher level of a botanical raw material, or perhaps a significant history of human use does not exist or may not be well established. This approach comes in handy. It has been published, as I mentioned. It's available as open access, and I recommend that you obtain this article for more details and more case studies that are presented. But in the next few slides, I will walk us through a few key decision points of this very complex diagram shown at the left here. So you begin here and with assessing what information you do have, and you get to a point where maybe there's some unresolved endpoint gaps, or different botanical extract preparations from established uses is your situation. Higher dose levels are identified. We ultimately get to this stage where it really requires phytochemical characterization and identification information. And we call that, constituent characterization and identification, or CCID. And as I mentioned previously in my description of the analytical approach that we use. So we get to a point where we have to have that information, and then we use that phytochemical constituent-based analysis of botanicals for rapid identification of those ingredients, those phytochemical constituents. And we begin to categorize them, and we can bucket them. For example, these phytochemical constituents are commonly found in the diet. And then we acquire consumption data, dietary intake levels, and we can quickly say, okay, the levels that we're going to be using in product are no different or less than what people are consuming in the diet. So we don't have to worry about this. So we can quickly, assess a number of those phytochemical constituents. And then the case study that I'm going to present today really illustrates how we use the phytochemical characterization information, and we take into consideration genotoxicity data and what we call in toxicology a threshold of toxicological concern, or TTC approach, to narrow down phytochemical constituents that may require potential further investigation. So briefly, the TTC approach involves the assigning of constituents to chemical classes and making worst case assumptions about their toxicological profile. The case study I'm going to use is Polygonum cuspidatum. So I love these pie charts that we develop because I think it's a very helpful way of quickly bucketing all of those phytochemical constituents and presenting them in a way that gives you, a quick, puts that botanical extract into perspective. So in this example, you can see that the majority of the botanical raw material is composed of maltodextrin, and that's in this case probably used as a carrier. And then a smaller portion of phytochemical constituents were bridged to food ingredients, so dietary intake, like I mentioned previously. So we can discount those, and we can cover those from a safety perspective. And then in this case, there was a significant component of the botanical raw material consisted of resveratrol, and then a smaller number of constituents consisted of various phytochemical classes, as shown in the box there to the right. So we can easily assess resveratrol, but what about all those other minor but potentially important from a safety perspective phytochemical constituents? So here we've broken those down. So that small wedge of phytochemical constituents, we get an additional understanding of the complexity of working with botanicals. Within each of these phytochemical classes, there are constituents that we then further identify, and then even more minor constituents that remain unidentified. And you can see that, the small, level of some of these. And for each phytochemical grouping, we can identify a total amount as well as individual amounts for the constituents within that group. And we use these data to then apply that worst case TTC approach and immediately rule out constituents that would require any additional follow-up. Now, to increase the acceptable TTC level to those lower level constituents would depend on availability of gene tox data for those botanicals or botanical constituents. With favorable gene tox data, that TTC threshold can be raised. And in our example, with this botanical extract, it really left only resveratrol and imodin that really required an in-depth endpoint assessment. If we wouldn't have had genotoxicity data, or we would have had positive genotoxicity data, the TTC level is so low that so many constituents would be above it, and it would require a lot of additional testing. So you would really be chasing, information down in the weeds. So we, in this example, could easily support the safety of resveratrol and imodin because they both have very good safety data packages. So lastly, when data gaps are identified for certain toxicity endpoints, we move to utilizing some of those 21st century toxicology techniques that we're looking at as part of the Botanical Safety Consortium. And this is to, adequately support the botanical in question. So it could include in silico approaches using certain databases that use, that contain structural alerts for various toxicity endpoints. It also uses in vitro testing approaches such as receptor assays and cell-based studies, some of which, as I mentioned, we're looking at in the Botanical Safety Consortium. So in summary, preclinical safety can be established through a robust history of safe use in the target population with consideration of sensitive populations, strong safety preclinical science that is grounded in full phytochemical constituent characterization, and the use of 21st century toxicology tools as needed, which show promise in supporting botanical raw materials. We have, myself and colleagues, a number of publications and presentations that we have done on our approaches to botanical safety. For myself, a lot of publications and presentations on botanical drug interactions, for example, a lot of the analytical methodology that I talked about, and other toxicity tools and approaches. And I have this in a brochure with hyperlinks. Most of these publications are open access, and I will share that with David and Helen for distribution to this group. And with that, I thank you and look forward to discussions or addressing any questions you might have.
Right. Thank you very much, Amy. Very interesting. So what's that? The resveratrol-rich plant, Polygonum. What is it used for? What are the indications for it?
That particular botanical, that's a good question. I was thinking
because I mean, you can just drink red wine, right? you can drink red wine, get nice resveratrol, and also get into good mood.
Right. That particular, botanical, I don't believe we have currently in a dietary supplement. I think we were looking at it, and I don't recall exactly what the product was, the anticipated product was, but I don't believe we're actually currently using that in a formulation.
Oh, okay.
We have at the USP, we have actually considered, we have looked at it a little bit, thinking about a standard, but I recall that there were some safety issues that made us not go forward with it. But I think one of the things that is mentioned is that it is used as an antioxidant, which is very broad and doesn't really mean much.
Yeah.
So thank you so much, Amy, for such a wonderful presentation. Lots of information. I'm sure everybody is looking to understand this more. I see a hand here. Somebody has one question. We can tackle at least one question before we move forward. Just one, and then we leave the others to the end. The name Tank.
It's Rose, or is it someone else?
I think Rose can ask. She had a hand
Rose.
up first.
Go ahead, Rose.
Thank you.
Go ahead.
I'm sorry, Amy, I really enjoyed your talk. My name is Rose Hayeshi. I'm from the Preclinical Drug Development Platform at Northwest University in South Africa. So I'm very interested because you indicated that your strategy is for dietary supplements, and we do preclinical rodent work for an industry academia, and we have a lot of people working with plant extracts for pharmaceutical use. And I was just wondering, I asked in the chat there, your TTC approach or this screening approach, do the regulators accept that for dietary supplements? And do you think that that is something that we could adopt for botanicals, where they're trying to use them as nutraceuticals or supplements for pharmaceutical use? Thank you.
We personally have not submitted in the United States. We would call it a new dietary ingredient notification, with taking a TTC approach. So I can't say that we presented that to the regulators and it's been accepted, but I would anticipate that that's a fairly common approach. It's well used in other regulatory environments, in EPA, for example. So I do think that they would be definitely be open to that approach. I just can't say from a personal experience that I have used that with the regulators. But I would not. I do know that the FDA is very interested in the work that we have done in this tiered approach, and we've actually been invited to speak to them at the FDA and give this talk. So I do think that it's well regarded and considered. So
yeah, thank you very much, Amy. I'm afraid we have to move on for the sake of time. Please hold your questions, and we may find some time at the end to answer some of them. So now I have the great pleasure to introduce our next speaker, who will tell us about about the application of botanical safety data in a regulatory environment. Our next speaker is Mr. Nana Buffy-Yeboah. Nana joined Health Canada in 2003 after completing his master's in biology at the University of Ottawa. He is currently a senior manager in the product assessment division. Previously, he served as assessment officer and head of unit, and Nana also served as a government liaison in past USP expert committees. His division at the Health Canada works on natural health products, which is what the equivalent of dietary supplements are called in Health Canada, in Canada. Specifically, they work on ingredients classification, the evaluation of license applications, reviewing clinical trials applications, monograph development, and health risk assessment. Nana will give us a view of the regulatory framework for natural health products in Canada. Welcome, Mr. Buffy-Yeboah. I give it back to you.